This is CGTN, China Global Television Network.
Hello and welcome to this Razor COVID-19 special. I'm Emma Keeling. In 8 months, we've learned that SARS-CoV-2 affects many different organ systems. Scientists have now added neurological manifestations to a lengthening list. Over 300 studies from around the globe have found a prevalence of neurological abnormalities in COVID-19 patients, ranging from brain inflammation, delirium, nerve damage and stroke. Patients suffering severe symptoms of COVID-19 may feel as if their body is under attack. Happening almost always at the same time as key parts of the illness are potentially fatal brain disorders. Robert Stevens, a professor of intensive care at Johns Hopkins Medicine, Maryland, says we could be facing a secondary pandemic of neurological disease.
I'm speaking about these, repercussions of infection with the disease. And some of which I believe could have, and many of my colleagues as well believe could have some long-lasting effects way beyond the acute phase of infection.
The manifestations are variable and in some cases life-threatening.
If we're talking about, acute onset of delirium or encephalopathy, this is affecting anywhere between 40 and 50% of patients with moderate and severe COVID-19 in hospitals. This has been shown in a number of different studies. If we're talking about stroke, ischemic stroke, this is being documented in 3 to 5% of people who are hospitalized with COVID-19. If we're talking about, infection of the brain, encephalitis, we think that this is much rarer. As for the other manifestations, things like peripheral nerve disorders and spinal cord, inflammation, myelitis, we think that these are quite rare.
Most of Dr. David Strain's clinical work with COVID-19 has been with older adults.
One of the key features amongst these individuals was an acute delirium. That is, they lost their awareness of their surroundings. They became acutely confused, more so than usual, even in the absence of dementia. And that was far more than we see from similar viral infections.
There are over 300 studies and counting into the neurological manifestations of COVID-19. Do we know exactly what is causing all these issues?
Patients who have the severe disease and also the more moderate disease experience very significant physiologic changes. So that means that their blood oxygen levels can go way down. They can experience very high temperatures. They may experience fluctuations in their blood pressure, and they have very prominent metabolic and inflammatory perturbations. And so we believe that those non-specific, physiologic alterations can profoundly affect brain function and may account for a significant proportion of the cases of encephalopathy and delirium. So that's the first mechanism. The second mechanism we think could be due to the immunologic response to the virus. The third mechanism is the hypercoagulable state. So we know that many patients, a very, very significant proportion of patients with COVID-19 have a tendency to form blood clots in their veins, blood clots in their arteries. The fourth mechanism is the one that I was alluding to earlier, which is the direct invasion of the brain, the entry of viral particles into the brain causing direct damage to nerve cells in the brain, as well as glial cells, and potentially an inflammatory response.
The brain is normally shielded by what's known as the blood-brain barrier. Throughout the body, there are small gaps between the cells which line the interior of blood vessels. These gaps allow ions and small molecules to pass from the blood to the surrounding tissues. But in the brain, these cells are closer together, even overlapping, which allows nutrients, water, and some gases to pass into the brain tissue, keeping out pathogens and toxins. But the SARS-CoV-2 virus is clever.
Things that cross the blood-brain barrier can affect the brain function, and we know that this virus does cross the blood-brain barrier. It's been clearly documented both in animal models and then in humans that it can transmit not just within the nerves of the brain, but also can cross synapses. So it can cross the connections between the brain which allows it to spread. In fact, that's possibly one of the reasons why the first symptoms is this anosmia, the lack of smell, that the droplets that are coming from other people will get either into our mouth or into the eyes. It will then enter through the eyes, down into the nose, and enter the brain through the nose before spreading around the rest of the body. So not all of COVID is coming in directly through the lungs.
So, I mean, does SARS-CoV-2 affect the brain differently from other viruses?
Yeah, so probably we're not sure that it does.
The influenza virus responsible for the 1918 pandemic known as Spanish flu caused permanent and profound damage to the brain. Patients developed delirium, encephalitis or inflammation of the brain, and there were delayed effects such as Parkinson's and encephalitis lethargica, also known as sleeping sickness. Coronaviruses can also infect nerve cells.
So this was shown in 2003 with the SARS, so the SARS-CoV virus was shown to be able to infect the brain and cause encephalitis. We also know that the Middle Eastern respiratory virus, the MERS-CoV, is also able to enter the brain and cause encephalitis. So it seems that coronaviruses have a propensity to enter the brain and cause neurological disease. Either that's one mechanism, but I think probably what we're going to find out, the prevailing mechanism is indirect, meaning not invasion of the brain, but causing brain disturbances through either physiologic changes or immunologic changes.
Have doctors missed the neurological manifestations in the brain from COVID-19 patients?
They have been reported on from the very beginning, from February, March, and up to this day. It can be very challenging, however, to accurately document neurological changes, especially in the severely ill patients who are, for example, in the intensive care unit receiving high doses of sedatives. It's, it can be, you can overlook even of a severe neurological problem in a sedated patient, and that's why I think we have to have a very high index of suspicion. I think, the physicians taking care of COVID-19 patients should be instructed to perform a very detailed and careful neurological history and examination, and immediately either seek consultation with neurologists or obtain additional testing such as imaging whenever there's a concern about a severe neurological problem.
Recent research has shown that T cells, which play a central role in our immune response, may last for years after targeting coronavirus. This is encouraging news after a Spanish study revealed that patients suffering mild symptoms of COVID-19 may only carry protective antibodies for a few weeks. It's the latest study to show that building herd immunity against the virus may be unachievable without a vaccine. But could our immune system have another plan? The true impact of the SARS-CoV-2 virus on the Spanish population has been a mystery. Over 28,000 people have died, but the government was unsure how far the virus had spread through the population. Now, a random study of 35,000 households across the country, testing over 61,000 people, has revealed only 5% of the population maintained antibodies to the virus. Professor Marina Poyan was the lead author.
And in those places more heavily heated, we have 10, 12%, 14% is the top. That means that we are very far away from the herd immunity that we are looking for, because specialists in herd immunity had estimated that in order to have this group immunity to prevent the spreading of the virus, we need 60% of people with antibodies.
Other experts have estimated that at least 70% of the population would have to recover from COVID-19 to halt the pandemic. But just because a person has antibodies, it does not mean they are protected, as immunologist Zania Stamataki explains.
We don't even know that the antibodies that people develop are protective. So this is something that still is out there, and if you have had coronavirus infection and you have detectable antibodies, we cannot say as scientists for sure that you're protected from reinfection and you're protected from severe symptoms. So we're still asking people to shield even if they have antibodies.
Antibodies are Y-shaped proteins which recognize other proteins called antigens. Antigens are molecular structures that may be present on the surface of a pathogen, such as a virus, which triggers an immune response when it enters the body. B cells have antibody receptors, which, if matched to the antigen, cause the B cell to bind itself to the antigen. Once bound, the antigen is ingested and broken down inside the B cell, with the resulting product placed on the surface of the B cell, known as major histocompatibility complex 2, or MHC2. Helper T cells bind to the MHC2 and release cytokines. The cytokines and T cell help cause the B cell to clone itself into memory B cells containing the same receptors, and also into plasma cells. The main function of plasma cells is to release antibodies. The antibodies created are specific to the antigen which was bound to the B cell in the first place. The memory B cells can last for years, and they can react rapidly in case of future reinfection. The study did not look at the neutralizing ability of the antibodies, but it did reveal that 14% of people who had mild symptoms and tested positive for antibodies in the first round of testing saw a decrease in the antibodies in tests done only a few weeks later. In a study by King's College London, the antibody levels of more than 90 patients and healthcare workers were monitored for 3 months after symptoms emerged. Tests revealed that while 60% of people had a potent antibody response at the height of the virus infection, only 17% retained that potency 3 months later. The study, that has yet to be peer-reviewed, found that antibody levels rose higher and lasted longer in patients who had severe cases. These results have implications for the development of a vaccine.
First of all, when you cleared the infection, was it your antibodies that did it? Are your antibodies sufficient to prevent you from having severe symptoms in the future? How much antibody is enough? And also the quality of those antibodies. So are these antibodies able to neutralize the virus from entering your cells? So the quality of the antibodies is important as well. To answer that question, you need to do laboratory-based tests, which are not readily available. The good news is that people that have had a lot of contact with infected individuals, so healthcare workers, for example, that may have had the virus and then they go back to work, we are not really seeing a lot of evidence of reinfection in these groups. So it is possible that they are protected. The data is a bit slim still for this, so we're waiting to get more good confirmatory assays.
As the world reopens, thorough testing of the population is crucial. For those who become infected but don't develop antibodies, the body has other defences. A Singapore study has highlighted the power of T cells, which are another immune cell. The research found SARS patients still retained their memory T cells 17 years after they were infected. So some virologists have suggested that, we're underplaying the importance of T cells, which can kill viruses. So are we focusing too heavily on antibodies?
We have found in patients that have recovered from COVID good T cell responses against the virus. So we know that the good news is you make antibodies and you make T cell responses. The next question is, how long do they last? Which one's better? We don't know. We don't know which one has contributed to clearing a virus. We don't really know that yet. Data show so far from other coronaviruses that your antibodies, for example, to SARS and MERS, they might wane after a couple of years, but T cell might last longer. So it is possible that you get a more long-term immunity from memory T cells that stay behind after your first infection, and they're able to act immediately following reinfection. For SARS-CoV-2, which causes the COVID-19, we don't know that yet, but these assays are now being done.
Life moves pretty fast. Ideas move at the speed of sound. Technology moves at the speed of light. If you don't filter out the noise, you might miss the details. We pay attention to the details because they matter. showing you a different perspective. See the difference.
Creativity, technology, and inspiration come together in a new series of podcasts from CGTN Europe. What ideas are out there for enriching our common future? Find out in This Way Forward, available wherever you get your podcasts.
While we all wait and hope for a coronavirus vaccine, many of us are keen to go about our day-to-day lives again. Physical distancing and washing hands have helped limit the spread of the virus, but can't prevent all forms of transmission, particularly indoors. With the virus known to stay alive in the air for a number of hours and on some surfaces for days, a physicist at Columbia University is harnessing the power of light in an attempt to stop the virus before it infects.
David Brenner has spent years developing an ultraviolet or UV lighting system that can kill viruses, including SARS-CoV-2.
UV light has been used to kill viruses for a very long time, and it's 100 years since we knew that UV light kills viruses. And certainly in the past few decades, people have been using UV light a lot to kill viruses. In hospitals, for example, it's used in operating rooms overnight. So when the surgery is finished, everybody leaves, and then you can use conventional germicidal UV light to sterilize that room.
Ultraviolet light is on the same spectrum as visible light, but has a slightly shorter wavelength that cannot be seen by the human eye. It can be broken down into 3 groups: UVA, B, and C. UVC is the shortest and most energetic, often called germicidal UV for its sterilization properties.
Conventional germicidal UV light is really good at killing bacteria and viruses, but you can't use UV light directly in situations where people are around because it's a health hazard. It has issues with the skin and issues with the eye. So what we started to think about is there a type of UV light that we might be able to use which actually still kills bacteria and viruses, but doesn't have the safety hazards of conventional germicidal UV light. And that's where the far UVC idea came from.
Conventional germicidal UV light has a wavelength of 254 nanometers, but is harmful to humans. Far UVC light has a shorter wavelength of 222 nanometers, which they believe is a lot safer.
So we've been working for a couple of years to look and see, does our technology work for killing influenza virus in the air? And indeed it does. So when the COVID-19 crisis came along, we realized pretty quickly that could be very well the same story, that we're looking to kill the COVID-19 viruses in the air while still being safe for human exposure.
Far UVC kills viruses by warping the structure of its genetic material, preventing replication. Professor Brennan and his team have studied the technology with 2 coronaviruses that cause the common cold. They found that 16 minutes of far UVC light kills 99% of coronavirus particles in the air. And they're confident it doesn't harm humans, which is supported by several long-term studies.
Essentially, on the very surface of our skin and on the very surface of our eyes is a layer of cells which are not living cells. So the far UVC light, as it's impinging on the skin, simply gets absorbed in those dead cells, and so can't reach the living cells in the top layer of our skin. On the other hand, bacteria and viruses are really, really, really small, and much smaller than human cells and groups of human cells. So the far UVC light can certainly penetrate them and kill them. And that's the basic science of why it would work.
Professor Brenner's lights are focused on efficacy and safety and are currently waiting for US Food and Drug Administration approval.
I think where our technology would be most applicable is simply where you have groups of people together. These far UVC lights will be part of the same structure as those visible lights, or there'll be a separate light hanging next to those lights with a gentle purple color, really to tell people that the lights are on and to reassure people that it's working. So many people would like these lights in all sorts of different settings, in offices, in hospitals, in schools, in restaurants, in theatres and airplanes. As we start to come closer together, we need all the weapons we can get to limit the transmission of the COVID-19 virus.
As some countries reopen their borders to international travel, airlines are implementing measures to reduce the risk of infection for passengers. A simple cough can project droplets of different sizes into the air. Gravity will force large droplets to the ground, but the smaller ones could stay suspended for a long time.
A study of a commercial flight in 1979 helps us understand how viruses can spread in confined spaces like planes. 54 passengers were on board and delayed for 3 hours before takeoff. At this time, the ventilation systems were not functioning. 72 hours after the flight, 72% of the aircraft passengers became infected with influenza, all from one infected passenger who had boarded the flight. Ventilation systems on most commercial airlines are specifically designed to circulate air. Air in the cabin is refreshed every 4 minutes through HEPA, or high-efficiency particulate air filters. Cabin air is blasted through a 3D mesh of microglass fibers to capture microscopic particles and deadly germs. The SARS-CoV-2 virus is microscopic, measuring approximately 125 nanometers in diameter, which is similar to other viruses like influenza. It's smaller than bacteria, which range widely in size, but start from around 1,000 nanometers and a fraction of the size of a red blood cell, or the diameter of a human hair. The gaps in the HEPA filter are large enough for a virus particle to pass through, but they're still caught by Brownian motion. Smaller gas particles are shot through the filter. These knock the virus particles around in a zigzag-like fashion, increasing the probability of collision with one of the mesh fibers, where it's then deposited. HEPA filters on board modern aircraft remove 99.995% of all virus particles, and the air is recycled through the cabin 20 to 30 times every hour. Regular maintenance and replacement of the filters is required to maintain that level of efficiency. However, even though the filters are nearly 100% efficient, within those 4 minutes, air has usually mixed across 3 to 4 rows in front and behind. Professor Chin-Yan Chen and his team have modelled different scenarios within air cabins to study how particles can circulate.
So you can see that the cough or talk, the droplet is go up to the ceiling level, then the ventilation system bring it down along the walls of the window and to the floor level, and then it could really go to the other side of the aisle, and then finally form this mixing condition.
Another well-studied flight on March 15, 2003, a Boeing 737 carrying 120 people flew for 3 hours to Beijing from Hong Kong, where a 72-year-old man with SARS had boarded the plane. The World Health Organization could only make contact with 65 people 8 days after the flight, but of those, 22 were found to have been infected with SARS. The majority of those were located close to the 72-year-old man, indicating localized infection. However, infected passengers were identified throughout the cabin, and it's believed they had no contact with the infected person, either before or after the flight. This has perplexed epidemiologists.
So in these journeys, this person infected more than 20 fellow passengers in the airplane, and then 4 of them died as a result. Which means the seat next to this sick passenger is a very high-risk seat. But you also see from this seat map, passengers sit as far as 7 seats could get infected. So we also suspect that the wake caused by a walking passenger or a walking flight attendant could bring these particles very far.
The challenge facing scientists in the aviation industry is understanding exactly how SARS-CoV-2 is transmitted from an infected person to another person, which is more complicated than you might think. We know that SARS-CoV-2 is primarily excreted through respiratory droplets which leave the mouth and nose. According to the World Health Organization, these droplets are relatively heavy, do not travel far, and quickly sink to the ground. These droplets can land on objects and surfaces around the person, such as tables, doorknobs, and handrails. People can become infected by touching these objects or surfaces and then touching their eyes, nose, or mouth. This indicates that the likeliest spread of the virus is from touching contaminated surfaces where the virus can remain viable for several hours and sometimes days. For these reasons, airports and air cabins are being deep cleaned and in some cases radiated with UV light to kill the virus. Strict hand washing regimes are also being promoted to prevent transmission. But this doesn't give the full picture. A recent hospital study in Singapore looked at SARS-CoV-2 viral particles in samples taken from the air surrounding COVID patients. They found that virus particles were present in droplet sizes ranging from 1,000 to 4,000 nanometers, despite the hospital rooms having the air change 12 times an hour. Droplets of this size can hang in the air for up to 3 hours, but limitations of the study meant they couldn't conclude whether these aerosol droplets were a possible route of transmission. However, one of the first animal studies of SARS-CoV-2 in golden Syrian hamsters has found that aerosol droplets was in fact a successful route for transmission. This controlled study is problematic for governments and industry wishing to reduce the 2-meter social distancing rule. If virus particles can hang in the air for up to 3 hours and continue to be infectious, then people need distancing and shielding to protect themselves. Ventilation systems must also not move infected air around. Professor Qingyang Chen and his team have designed a new ventilation system that doesn't mix the clean air in the cabin. Rather than from above, it's supplied from below, directly to your breathing zone. Then when you breathe out, the contaminated air rises and is extracted at ceiling level.
So as a result, you are not sharing the air with your neighboring passenger. With our new design, we anticipate that we could reduce the risk of infection by about 50%. Something I hope that the airplane manufacturers are willing to implement.
So when we're back on planes, how will things be different?
As you can see, most of US major airliners are asking passengers to put masks on. And some of them also leave the middle seats open. And I think this is a very important first step for the airliners to take those measures to mitigate the risk. I have to say that nothing is bulletproof. When you travel, you always have a risk. And risk is not only occurs in the air cabin. It could occur in the terminals, could occur in the buses or the subways. We hope that the airliners will take proper measures as I have described here. And this will be very important for building the confidence on the passengers.
And one airline chief executive has told CGTN that in the future, if a vaccine is developed, we may need to carry health certificates before we board planes. That's it for this COVID-19 special. Look after yourself and each other, and we'll see you again next week.